Good day, everyone, and welcome to Teach Me Ag with Miss Lindo. So today we'll be learning about plant growing medias. Now, if this is your first time knowing about those, that's okay. Follow along as we'll be discussing them. But first things first, let's see if you have prior knowledge about this topic. I want you to think about growing medias. Hmm. What's so interesting about green medias? So here we have a bell ringer to help get the juices flowing. Why do some plants grow better in certain soils or mixtures than others? I want you to think about time you planted anything, okay? Or even something. And what did you use to grow it? I'm gonna give you some time. Want you to think about that question and then you can resume this video. Alrighty then. So I guess you have already finished the question for the bell ringer. Hooray if you have done so. If you have thought about soil or maybe hydroponic systems, that's okay because this video we are going to get in the nitty gritty. So before we get into the lesson, we have to look at our standard, which is standard 4. Students will demonstrate greenhouse crop production techniques. Now this standard is coming from our precision floriculture blueprint. This is where I'm getting all the content for this standard from, okay? All right. When it comes on to our learning intention, we're looking at objective 4. For objective 4, we're going to examine the components and the properties of growing media. First things first, we want to make sure we are able to describe the components of growing media. Secondly, we want to be able to describe the function of growing media. Third, determine the desirable properties of growing media. And last but not least, evaluate the advantages and disadvantages of soilless media. So let's move forward. So we already got the standard and learning intention, but let's break it down even more in smaller chunks in terms of our success criteria. First things first, before we can talk about describing any components, let's talk about be able to define the following. One, we should be able to define what's a plant growing media or growing media. What's soil? What's soilless and mixed grain medias. Another thing, you and I should be able to describe the components and the properties of grain medias, identify the categories and the components of these medias, evaluate the medias based on plant needs, and last but not least, compare the pros and cons of soilless media. And here are key terminologies that we'll be looking at throughout this lesson. And you can pause and go down the list, but you're going to hear them a lot. All right? I'm just taking a look. I'm wondering what these white things are in this picture with soil. I look like a cactus plant. We'll know in a second. All right, so first things first, what's a plant growing media or a growing media? Basically, with plant growing medias, we're looking at the basics. These are the essentials that are providing nutrients, they're providing support and water to the plants. So with this, it's going to help with overall health and growth. Of course, with a nice and stable environment, those plants are going to flourish as long as they have everything that they need to survive. The material in which plants are grown. It also helps the plants to support its roots, provide water, air, and nutrients. You must be wondering why we're talking about air, water, and nutrients. With all living things, they're going to need water for hydration, air for respiration, and nutrients in this case for plants, aka their food. in order to survive. So here we're going to look at the components of growing medias. Here are a few components that all plants need in order to survive. We have organic matter. The simplest way to think about it, think about a time where you may have, unfortunately, a loved one that passed away. You bury that loved one. Over time, their body decay. So there are microorganisms that helps to break down the flesh, the bones, the organs of your loved one. And over time, your loved one, or it could be a human or animal, will become organic matter. It's the same that happened with plant leaves as well. Or think of poo, animal poo too. This happens as well. So in this case, with animal matter or organic matter in general, sorry, not just animal, can happen with plants too. Overall, what happened is that it's going to help with improving water retention and aeration. You may be wondering, what is aeration? Hmm. So with aeration, this deals with the circulation of air within the soil. Let's look at another components. We're looking at minerals. In this case, minerals can be inorganic matter. So we're thinking of sand, silt, and clay. Sound familiar, right? We'll talk about that in a second. And it provides structure and drainage. So without these three, we wouldn't be able to have a good structure that can hold our plant in place. And not only that, help the plant to get rid of that extra water. Because even though water is good, too much water is not good for that plant. Air. Why do plants need air? You may be wondering, why do plants need air? Let me remind you again. When it comes on to air, it helps with respiration. Respiration means breathing. Do it with me. The plants do the same thing. They breathe. So, in this case, within the plant cell structures, there are spaces. There are spaces where the air is going to flow. In order for air to get into that plant, air goes through the roots, through the small pores in the roots. And also, there are small pores in the leaves called stomata that helps with the airflow as well. And of course, with airflow in this case, if we have air going through the growing media, we're helping to get rid of the extra water as the water evaporates in the atmosphere. Yay! Water. H2O. Tyler did say water, but we ain't talking about that type of water. So water, in this case, helps with hydration. And not only help with hydration, not only help with holding the pores, hmm but also help with dissolving them nutrients, aka food plants, taking them up, racking them up, leveling up. Now, the next thing we're looking at is nutrients. You may be wondering why she keeps saying nutrients, nutrients, nutrients. Remember, nutrients is plant food. Plant food. Think of it this way. When you don't get food in your system, you become sluggish, you become an angry man or woman. It's the same with plants. If plants don't get their food, they gonna act up. And same with water. If they don't get their food and water, they gonna act up. They're gonna start wilting. They're gonna start looking funny. They're not gonna grow. So with nutrients, aka food in this case, it provide the necessities, the elements like nitrogen, phosphorus, potassium for that plant to grow. And if you have ever seen a fertilizer bag, you may be seeing numbers like 10, 10-10. That's like a small version of showing NPK, aka in this case nitrogen, phosphorus, and potassium. I'm not going to go any more details into that right now, but just a surface. And last but not least, microorganisms. So the word in this case, micro, refers to small, and organisms refers to living beings in this case, living biotic factors. So with these small microorganisms, guess what they're gonna do? First things first, even though they're small, we're talking like bacteria, fungi, earthworms, they are gonna help with breaking some of those extremely complex nutrients. They're gonna break it down into a simple version so that plants can absorb all of them. Do you get what I'm saying? If not, let me break it down further. Think of your own body. When you eat food, it has to go through that digestion process. You have different parts of the body helping with the aid of digestion. It all starts with the mouth, right? And the acids and all that helps to break down the food. Think of it this way, but the microorganisms is doing it for the plant, so the plant roots can absorb everything. As I know this is a recording, at any time you can rewind, watch this video so it can help. Now the next thing we're gonna look at is the function of growing medias, and we should be able to explain them. So I kind of went into the nitty-gritty of them when I was going through the components. The key functions of these growing medias are plant growing medias. One is to hold those roots in place. So any growing media that you have, it should be able to help that plant root stay in place. They feel stable. Think think about this. Think of part this part of the plant, the root. Think of it as the feet of that plant. It helps that plant to stand up. It has all the support it needs from its feet and legs to help it get up. Think about it that way. Also think about with these growing media, it provides nutrients. If the media wasn't there as a foundation, this plant would not survive. It needs something to give it its food. It needs something to help it stand. It needs something to take care of it, to retain that moisture that's coming from that water, to keep it hydrated so it can survive in, for example, in extreme temperatures right now, like how hot it is right now during the summer. It needs to help to keep some of that moisture so that plant can have access to put at any time it wants. It needs to have that flow of oxygen and have that exchange of oxygen and carbon. And where is that coming from? It's coming from the atmosphere. It's coming from the water and the nutrients once they're broken down into simpler, complex atoms or molecules or elements. And they also help to support those microorganisms as we mentioned from the slide before. And of course, we have illustrations here showing that. All right, moving on to the next slide. So here's another exciting part. So maybe all your life, maybe in your case, the only growing media you knew of was soil. Thanks to modern technology and thanks for people learning and studying that there are different ways of growing plants with and without soil. We have three, some people may see as three major characters, some may see as two, but I wanted to include all of them. So we're going to say three major categories of plant growing medias. So the first one is considered as soil based, the second one soilless, and the third is mixed media. So soil based, As the name pertains, as a preview, think of it as the good old natural soil that you walk on every day. That you see outside where the grass is growing, the flowers are growing, the trees are growing. So let's think of not soil, as the name suggests. And then mix, hmm, mix is like a mixture of both soil and soilless, giving the best outcome for that plant to grow. And of course, we have illustrations matching with each of these different types of soil of growing medias. Now we're going to go into the nitty gritty. So, as it pertains to soil based medias, as mentioned before, this is its natural form. So, when it comes onto soil based medias, we have organic and inorganic materials that provide essential nutrients and structure for plant growth that may help with the challenges of drainage and pathogens. So a lot of times, depends on where you live, you may have seen the following different types of soils. We have clay, aka I'll hear this a lot, red dirt. No, it's not dirt, it's soil. With dirt, you can't grow anything. Nothing can grow there. With soil, it's alive, and we're able to grow plants. Okay, so that's the difference. So we have clay soil, loamy soil, sand. You may see this at the beach, and silt. So let's look deep into different types of soil media. First things first, we have silt. When it comes onto silt, it tends to be silky, it tends to be powdery. And of course, with this is like a review video, but as a class, we'll be exploring the textures of these. So you guys will have an opportunity or already had the opportunity to feel these for yourself. Feel the texture, witness the water holding the drainage, and just write know what you observe. Of course, so the particle size that will be given by your teacher, which is me or any other teacher who may use this video. When it come on to the water retention or water holding, it's very high. The drainage is moderate, so it's in between. And then last but not least is in terms of its size, it's considered small and it tends to be fertile and smooth. So you will normally see this nice nice soil that is very silky and smooth, prone to erosion, and it retains moisture longer. So it holds on to moisture longer. The next type of soil that we're looking at is clay, aka we see as sticky, we see as smooth. When it comes onto water, it holds onto that water for a long time. However, this may some in most cases, this may not be the best to plant plants in, just because even though it holds onto that water for so long, the drainage is very poor. So it's just a few drops of water that come out. The rest is just held on in there tight. It holds on to that nutrients well. It holds on. It compacts easily, and it gets hard for those roots to penetrate. And actually, can give an example because the other day, some of my students and myself, we were planting plants in our courtyard at school. And at the time, I didn't know it was clay soil until I started digging, and I'm like, oh my god, my arms hurt. It wasn't just me, everybody was complaining. Our arms hurt because that soil was so hard. It was so hard, and then we're like, oh no. We were able to manage to dig those holes, plant the landscaping plants. But it just to give you a representation, and also what helped us to know that it was actually clay soil was that prior to digging, there were some areas in that courtyard where the rain had fell a day or two prior, and you could see evidence of the water still there because the water is just floating out top. It retains that water, it's holding on to that water, it's not letting go as much water as it should. And of course, because of the retention of water, the particle size in this case is going to be very small. The more it drains water, the bigger the particles, the less or the least it drains water, the smaller the particles. Loamy soil, that tends to be everybody's favorite. When you take it up, it tends to be crumbly. It tends to be soft. It's a moderate to high water retention. Is very good. It's like besties for a lot of gardeners and plants, just because not only does it take in that water and store some, but it let go the rest. It's very ideal for most plants, as you can see at the notes. It helps with that balance between all different types of soil, which is silt, clay, and sandy soil. And of course, it has mixed particles. So it's like medium size, in other words. Medium size, we're holding that water and we're letting go that water. No sandy. Sandy, I'm not saying you can't grow any plants from this. However, it it's a complicated relationship, and you're gonna see why in a second. So with sandy soil, it's very gritty, it's very coarse. The water retention is low because the minute you have a plant and you water that plant that has sand as its growing medium, that water is going to go through. It's going to go through. If you have ever been to the beach and you have sand on you, that water just go right through that sand. It's very excellent, as I just mentioned with those examples. Very excellent in terms of drainage, and it's quick. It drains quickly. It holds. This is a problem though, because it doesn't hold that food very well. So it's low nutrient holding, and it warms up faster in the spring. And of course, because water just goes shoop through it, the particle size is huge. Maybe I'm over exaggerating, but it's large. Now, here's some individuals' favorite. Here we have mixed growing media. So mix is a combination of, think of it as a combination of soil and soilless media. That's how we can think of it, right? And it offers that balance of properties. You have the benefits of the drainage that you need, the water retention you need, every support that plant needs for its development. So why people use it? It's the best of both worlds, as just mentioned in the example. I mean, in the definition. For example, with potty soil, we have a mixture of peat moss, perlite, and we have soil. You plant that plant, the perlite, as we're going to talk about it later on in the presentation, the perlite, it helps with drainage. The peat moss, it helps with retention. The soil, nutrients, retention, is giving the benefits of both worlds. And then soilless media. Soilless, simple as it is. I know it's not in this picture. I'm so sorry. Well, it's not, you can't really see that well in this picture. But with soilless media, think of it this way. It doesn't involve soil. Let's start with that because it says soilless. Soil is absent. So in this case, we're creating a sterile medium environment where plants are able to grow from organic and inorganic materials. All right. So with this, these materials are often used with fertilizer to make sure that that plant is receiving every food, nutrient, minerals, or plant food, nutrients and minerals that it needs to thrive. And there are two types, as mentioned. We have organic and inorganic soilless media. And here are a few examples. We have coconut coir, which we're going to look at those later on, the different types, as well as peat moss. Organic. We got to know what organic is before we can talk about the types of soilless media. So, organic, from this day moving forward, if you do not know what organic is, that's okay. We're going to learn it right now. So, when it comes down to organic, we're thinking of natural materials that are broken down. So, when it comes on to soilless media, organic soilless media, they are derived from materials that were once living organisms. Living organisms, aka biotic factors or biotic organisms. So basically, they're typically rich in organic matter and they often provide good water retention and some nutrients content as they decompose. So we're looking at example like peat moss, coconut coir, bark as in the tree bark, sawdust, compost, and rice hulls. So they're going to break down over time. Inorganic soilless media. With inorganic, we're looking at the compost of non-living organisms, aka abiotic factors, mineral base, and also mineral based materials. So with these, they do not decompose, so they tend to last longer. They're they're sterile, they're lightweight, and they're usually good for proper aeration and drainage. So we're looking at rock wool. Rock wool is very popular in hydroponic systems, and perlite, which can also be used in hydroponic systems, and some people use it in soil mediums. So here are the different types. And I know this video is a bit long. As I said, you can pause, come back, watch certain parts of the video. So here are the different types of soilless media. So we're looking at perlite. Sorry, peat moss. So peat moss, in texture wise, they tend to be soft and spongy. Kind of remind me of them sponge that you use to wash dishes at home. That's also the spongy texture. It holds water at a high rate. In terms of drainage, it's low to moderate. So it'll hold that moisture, and of course, it's gonna give it some time, and then of course that moisture is gonna evaporate because of its atmosphere. And then last but not least, it tends to have acidic pH. It's excellent moisture, has excellent moisture retention, and it's low in nutrients. So of course, with this, meaning that you have to provide nutrients to it, it's not going to really give that nutrient that the plant really needs at a certain level if those nutrients are not added with fertilizer. Perlite. Perlite is gritty. Think of small rocks. That's the best way I can describe it. It's gritty. It's so lightweight. Sometimes, in most cases that I've seen, or it could be the only case, I've only seen them white. Think of the small rocks, right? When it comes on to water retention is very low, drainage is high. So if you notice, anytime the water retention is low, the drainage is going to be high. If it's in between, the drainage is going to be moderate. So in this case, it's going to help with that proper airflow that the plant needs, what the root needs. Another thing you need to know about perlite, the way how they are made, is made from volcanic glasses, and they also add to that drainage to those mixes. So like the mixed medium, it helps with that. So at our school, we use soil and we use perlite because we know that soil is gonna help to hold them nutrients, hold that moisture, right? Which I forgot to say, we use loamy soil in our greenhouse. Um, in that case, with the perlite, it helps with that drainage. So we're not having that much of a waterlog situation. That water is coming out. That waterlog happens when you water too much. In other words, coil, aka coconut coil or fiber, it tends to be fibrous. If you have ever seen those, it's soft, it's moderate to high in terms of water content or water holding. In terms of drainage, it's moderate and its pH is normal or neutral. So when we talk about neutral pH, which we'll talk about further, but just to give you a preview, we're talking at 7. It's not acidic, it's not alkaline. And a sustainable alternative to peat, as in referring to peat moss, and it's good for water retention. Vermiculite, a lot of people use this for hydroponic systems. Vermiculite, it tends to be flaky and soft, sometimes look gray in color. It's very high in water retention. It's moderate in drainage, and it holds up water and nutrients. Used for seed starting and cuttings. Rock wool, I mentioned this earlier. Eh, rock wool tends to help with hydroponic system. So rock wool is fibrous, it's dense, it's very high in water holding, drainage is moderate, and then with this, it's used in hydroponic system, as I mentioned before, is sterile. It's excellent for water retention, and it needs pH adjustment. So if you don't adjust that pH, one may it could be acidic in that hydroponic system, next way it could be alkaline. So you need to make sure that the pH level is appropriate for that type of plant that you're growing in your hydroponic system. And then last but not least, not everybody uses it. Some people use it, some people don't. If they do use it, they don't use rock wool. Or if they do use rock wool, they don't use the clay pallets that I've seen. If you have seen something else, please let me know. So the extended clay pallets, basically they're round and hard. They tend to be red, suggesting clay. They are low to moderate in terms of water retention. They're very high in terms of drainage. And then last but not least, they can be seen as reusable, sustainable. They're sustainable, they're sterile, and they're excellent for hydroponic system and aerations. All right, so let's check for understanding. So first thing first, the first question that we have here, which of the following is considered organic matter in growing media? Let's think. You can pause the video and then we can come back to the answer. Number 2, black is essential for root respiration and fills the pores in growing media. Number 3, true or false? One function of growing media is to hold plant leaves in place. And last but not least, number 4, which of the following is not a primary function of growing media? Is it A, supports microbial life, B, provides nutrients, C, blocks sunlight, or D, retains moisture? So as I said before, you can pause the video so you can answer these questions and then we will resume. All right, let's go over the answer for number 1 as it pertains to organic matter. So I'm using my highlighter. As it pertains to organic matter, your answer is going to be peat moss. Remember, peat moss is an organic matter that breaks down. It comes from a living organism. With the others, they're considered inorganic matters. So with that, it's going to be there for a while. Doesn't necessarily decompose or break down. So that's why we said peat moss is going to be the best option for question one. For question two, as we talk about root aeration and helps to fill in the pores. I wonder what that could be. Root respiration. I think I just gave the answer. So the answer for this is gonna be aeration. I'm sorry if the writing doesn't come out cute. And that's okay. Remember, aeration is gonna help with those roots getting the air that they need. The air is coming in, the air is coming out. Not only is gonna help that plant, I'm trying to draw this plant, but it's gonna help those microorganisms that are around those plants. So those could be worms, and there's my little worm right here. Those could be bacteria. That's my bacteria right there. So that's what we want to think about. Now for number three, the true and false. One function of a grain media is to hold plant leaves in place. Are we really trying to hold the plant leaves in place, or are we trying to hold the plants? Hmm. Hmm. Nah, we ain't trying to hold them leaves. Yeah, so leaves gonna be held, but in terms of grain media, nah, that's gonna be false. We gotta clock that T, right? So that's gonna be false. Remember, when it comes onto grain media, it's not gonna help to hold those plant leaves necessarily. So we're gonna scratch that, but it's gonna help to hold the plants with its roots up. So if you have those roots that are anchored very well, the thing that's gonna happen once those roots are anchored in that growing medium, it's gonna help with the plant support. So once the roots are anchored, the spine of that plant is gonna be supported or as well. That's gonna help to support the entire plant, not in just only structure, but also making sure those nutrients, those minerals are flowing along with the air and water throughout the plant. Sorry. All right, then number four, which of the following is not, let me underline this, because a lot of times we skip this part, which of the following is not a primary function of growing media? A, does it help support microorganisms? Yes, it does. So the not part, that's going to be O. Does it provide nutrients? Yes, it does, in terms of our plant growing media. Does it block sunlight? No, it doesn't. Does it retains moisture? It does. So the correct answer in this case will be dun 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 dun. It's gonna be C. Which plant growing media blocks sunlight? I don't know any. I do not know any. So if you had those answers, you can. I'm sorry, I kind of erased them before, but you can always rewind and check to see if your answers were correct. And we're gonna move on to this second part of this unit. All right. So now we're moving on to the desirable parts and properties of growing media. So when it comes down to growing media, we want to look for the best of the best that our plants can thrive, they can flourish, be the best in their golden years, right? So first things first, we want to make sure that one, our plants are having, and I'm sorry if it's a bit tiny, but they have and they're being provided with adequate drainage for those roots. Because remember, with adequate drainage, those roots will be able to erode, aka aeration, helping that plant to grow healthy and strong. They need that air. With less moisture, with less overwhelming moisture, that plant is able to do its thing. When it has too much water, those poor plant cells, and I know if you have never heard of plant cells before, and this may be your first, that's okay. Where every living organism, they have cells. With plant cell, because of the way how their outer barrier of that plant cell is, if it consumes too much water, that barrier is gonna break. It's not like animal cell that may adapt to the additional water. With plant cell, it's not gonna do that. When that when that cell wall, that's the word I was looking for. When that cell wall breaks, game is over with. Though the plant is poor plant is gonna suffer, it's gonna die. So we don't want that to happen, okay? I want to always make sure that we're maintaining that proper water retention level. In addition, we want to make sure that we are having a balanced pH, the best of the best for our plants. With proper pH, we're able to have the best nutrient absorption and consumption. Two, as mentioned before in previous times, we want to make sure we're keeping it free. We're keeping it free for our living organisms to help those microorganisms to help break down the nutrients. But also in this case, we're also making sure they're making it a space where we're not gonna have these nasty pests. And pests, think of pests as the ops, the enemy. They're coming there to destroy your plants. And we don't want to give them that opportunity. So we want to make sure that we're creating um, a free zone, a harmless environment for that plant. So these pathogens and these pests don't come and harm our plant. And I also want to make sure it's lightweight, easy to handle and transport. All right, let's get to the exciting part of the soilless media. So there are pros and cons to basically think about it for anything that's out there. So the pros, to me, the pros kind of outweigh the cons, in my own opinion. But you will see for yourself. You can decide whether it's this is something that you want to do at home in your own comfortable space, especially living in an urban area. So here are the pros of solid media. The pros, it provides excellent drainage and aeration for that plant. It helps to reduce the risk of soil borne diseases and pests. It's lightweight and easy to handle. It's consistent and uniform in composition. It's ideal for controlled environments like greenhouse and hydroponic systems. And then last but not least, is easy to sterilize and reuse depending on the type of material that you're using. Now, in terms of the disadvantage, of course, it's going to require frequent watering due to low water retention. It may lack natural nutrients. So, in other words, you would have to provide it fertilizer or nutrients in order for that plant to grow. It tends to be more expensive than soil, which makes sense because you don't necessarily have those um, microorganisms to always break down the nutrients that are being provided. It may dry out quickly without proper monitoring. So, for instance, one of my students, he started on the hydroponic system, and I didn't say anything to him. I let him do his own thing. Of course, there were hints I kind of dropped to say, "Hey, check on your hydroponic system." He did not give his hydroponic system any nutrients for 2 weeks. Didn't check on the pH. And of course, unfortunately, the plants didn't do that well. And then it's not suitable for all plants. And then last but not least, oh, by the way, before I go to the last part, what I mean it's not suitable for all plants. For example, if you have plants that are grown provision plants, they not they may not grow well in a soilless media versus if they were to go in grow in soil medias, like maybe sweet potato could do well, right? If someone had tried it, please let me know, because we are always learning every day. Or you could think of Irish potatoes, for instance. And then last but not least, less buffering capacity for pH. And of course, the nutrients are going to fluctuate. So if you don't put the necessary nutrients and check on that pH, it's going to be all over the place. Which is a disadvantage of soilless media? Is this A, it's lightweight and easy to handle. B, is uniform composition. C, dries out quickly. Or D, reduces soil-borne disease. In the meantime, while you're thinking about it, I'm moving on to the next questions to read so you can have these in mind before we answer them. Number 8, soilless media are often used in blank because they're sterile and provide good drainage. And then last but not least, true or false? Soilless media require no added nutrients. So at this time, you can pause the video, you can answer those questions on your own, and then we can come back and discuss the answers. All right, so let's go over the answers. So as it pertains to the disadvantage of soilless media, let's elaborate and look further. I'm gonna choose a nice color so we can see. All right, so lightweight and easy to handle. Does that sound like an advantage or disadvantage to you? That's what I thought. That sounds like an advantage. So that is gonna be uniform composition. Does that sound like an advantage or disadvantage? You said what? Sorry? You said advantage? Same. I believe so. Dries up quickly. Does that seem like advantage or disadvantage? Not quite sure yet, right? So let's look at the last option that we have. It reduces soil-borne diseases. So does that seem like an advantage or disadvantage to you? Seem like an advantage, right? So our only option that is left would be dries out quickly. So that's the correct answer right here. So for number 8, we're looking at soilless media are often used in blank because they're sterile and they provide good drainage. Hmm. I'm I'm thinking the same thing as you're thinking, right? Yeah. So, soilless medias are often used in hydroponic systems. I'm sorry if my handwriting is not the best. I don't know how to write with this hydroponics, right? So the reason why we say this is popular. This is like the urban way of farming. And a lot of people are able to grow their food at home, either at a large scale or a small scale, and even businesses too. And then last but not least, true or false? Soilless media require no added nutrients. Is this true or false? I want you to really think about this one. Because when it comes onto soilless media, remember, it's soilless. With the soil, we normally have those nice microorganisms breaking down, any materials that may be above surface or below surface. It's breaking those materials or nutrients into smaller molecules or elements so our plants can absorb it. But what if your environment doesn't have any, right? So we're going to look at that. And for this, oh, you said false? Yeah, I bet. Yeah. So false is the correct answer. So in this case, false is going to be the correct answer, as with soilless media, they need additional nutrients. Because the statement said it requires no. So that's false. All right? So we're going to move on to our next part of the unit. At this time, if you like to pause and come back another time to watch the video, that's super. Otherwise, you can move on. So before we move on to the other part of this unit, let's look at the key takeaways. So real quickly, we looked at the components of grain media. We looked at the functions, the desirable properties, and last but not least, the soilless media pros and cons. And of course, it's not on here, but we did look at the types of grain medias, plant grain medias, right? And we look into the subtypes as well. So first things first, in terms of the components, we notice with growing medias, they have to have some form of support. That plant needs support. It's going to also need the air, it's going to need water, it's going to need those nutrients, it's going to need microorganisms. And even if it doesn't necessarily have the microorganisms, it's going to need something to help break them nutrients in a simpler, complex form for that plant to take up, right? So that plant can have the enriched nutrients, the enriched environment that it needs to thrive. If it has a positive environment filled with positive things, it's gonna do its best. Clap that T, as my kids would say. Then, for another thing we have to look at, in terms of its functions, pertaining back to its components, these growing medias must have something to anchor those plant roots. It must retain those waters and nutrients that that plant needs. And it also needs to have some form of air going through, providing the best air, the best carbon, the best nitrogen, the best oxygen that that plant needs for respiration and also for nutrient consumption, right? And it also helps to benefit those microorganisms that are around. It needs to also help with the desirable properties. So you want to look at the desirable properties in order to know what type of growing media that you may need to choose that is best for your plant. So we're looking at proper drainage. As you, if you notice these terms, these key terms I keep repeating, right? You're thinking of proper drainage, we're thinking of aeration, we're thinking about the water retention. That pH is necessary. If it does not have the correct pH, that plant is going to that plant is going to say bet and clock out. That's exactly what that plant is going to do. I want to make sure that we're not having anything harm that plant. That could be pests or pathogens. We want to make that lightweight and easy to handle. And last but not least, the pros and cons. Want to make sure in terms of our pros, we're looking at what's best for aeration for that plant to receive the necessary air it needs for respiration and also carrying out any other parts that it may need for that plant to thrive and produce, which also kind of, if you think about it, kind of help with photosynthesis as well. But that's another topic for another day. It helps to reduce those diseases and risk. And then in terms of the cons, sorry, it helps to have low content, nutrient content. Well, not necessarily help, but doesn't help. That's the correct word. It have low nutrient content. It dries out quickly. Um, it's going to be costly because you have to monitor it. And we're thinking about systems like a hydroponic system. As I said earlier, we did talk about the definition, what a growing media is, but these are like the major, even though that's major, these are the major takeaway of that unit. If you have any questions, feel free to ask, and you can always comment below the video or ask me or any teacher who may use this material. Now, if you are hitting the pause for this or the end, that's cool. If not, we're moving on to sampling techniques for growing media and testing pH. So the standard remains the same because this is all under the same standard, which is standard 4. We already went through those. If this is your like part 2 of watching this video, that's okay. you can also read from the slide, but I'm going to skip over it. In terms of our learning intention, no, we remember for the learning intention for the first part of the unit, we're looking at examining the growing mediums. No, we're investigating these chemical properties of the growing mediums. So you can read, you can look at different parts that we have to look at, and I'm just going to summarize. So at the end of this unit, basically, we need to demonstrate how these techniques are going to help with in terms of sampling these growing media to see what's best for us. We're going to test, we're going to determine the pH level of these various growing medias. And last but not least, we're going to interpret the results that we gain from testing these growing media samples. The success criteria is similar. In this case, we're actually going to do it. So the learning intention is telling us what to do, and the success criteria is saying at the end of this lesson, we are going to do this. So we are going to demonstrate proper techniques for sampling growing medias. And last but not least, we're going to test and determine the pH level of various growing medias. These are the key terminologies that we're going to talk about throughout the lesson. You know, most importantly, we have to give thanks. We have to make sure that we are knowing what pH is, because since it's the start of the show, we have to know what it is, all right? And we have to know what these terminologies are really are, and which we're going to discuss throughout the unit. So sampling techniques. All sampling techniques is basically collecting using different methods to collect samples or portions of growing medias so we can accurately test or represent that whole media to analyze and see what the pH and nutrients and other properties are of that medium. So when it comes on to testing that media, first things first, before we can test it, we have to talk about that proper technique. So we want to make sure before testing, we're going to collect those samples. We want to make sure we have all our equipment, they're clean before we go into different zones and collect. So for example, in this case, we're talking about soil. So we're going to look into the area where we're collecting that sample. We're collecting from multiple spots, so we're able to get accurate representation. And I'm just summarizing what's on the slide. Once we collect from different areas, we're able to combine those samples to create a uniform composition of that sample. We're going to label and store it. We're going to make sure we label it properly. Then when it's time for testing, we're able to test. And anytime you're collecting samples, we want to test as soon as possible so that we're able to get the accurate reading of that pH or nutrients before anything changes. So first things first, before we go in depth, let's check for understanding real quickly. So when it comes on to first things first, collecting samples, we're going to look at all of these. Which of the following is the correct depth to collect samples from the root zone? That's question number 1. Question number 2. Should you collect samples from multiple locations? Why should you collect them from multiple locations? You can pause the video and then answer the questions, and then we'll go over the answers. So for number 1, as it pertains to the correct depth, do we want to do 6 to 10 inches? Would that be necessary? Nah, that seems too deep, right? That's what I thought as well. So I'll say, nope, that's definitely oh. What about 1 to 2 inches? That seems too near. That's something like the organic matter. I'm not getting into the nitty-gritty, right? So let's accept oh. What about 2 to 6? We'll think about it. What about the top layer only? Top layer is like thinking of the first, maybe the first inch, right? So maybe not, right? So our correct answer will be dun, 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 it's gonna be C. So we're looking at 2 inches of collecting that sample. All right, number 2. Why should we collect the samples from multiple locations? Is it to reduce the time spent on testing? Nah, not really. Is it to create a uniform and representative sample? Maybe. Is it to increase the acidity of that medium? Right. We don't even remember talking about that, right? So let's take that out. And then last but not least, is it to create the sample heavier? That don't even make sense, right? That's what I thought too. So it's gonna be B. Because we want to have an accurate representation of what we're testing. Let's go to the true and false section. For question number 3, is it okay to reuse tools from previous testing without cleaning them? True or false? You said what? No, honey, it's okay. It's okay. We have to clean our tools because if we don't, it's going to affect the results. Think of it this way. And I know it's slightly might seem off topic. If you were doing surgery, right? Would you want the doctor to use the same tools that he used on someone else without cleaning them on you? Right? Think about that in terms of us testing our sample. So in that case, this is going to be false. So we have to clean them. We have to clean them. All right, question number 4. Sampling should be done immediately before testing to avoid changes in pH or nutrients. Sampling should be done before testing. Yeah, yeah, that's true. Want to make sure we have to know what we're testing. So we have to collect those samples first before we can test, right? And then last but not least, you only need one sample to accurately analyze a growing media type. Question 5 seems very familiar to question 2. So if you answered question 2 correctly, you already know we're not going to need one sample. So that is going to be false. You did an amazing job. All right, so now that we went over the sampling, let's get into the testing and determine the pH level of various growing medias. So this right here is just an example. Excuse me. This is just an exemplar of what a lab would be like for a growing media, various growing medias. So first things first, I have an example of what we'll be doing in the class. So we have our objective. In this case, we're going to determine the pH level of different types of growing media and evaluating how the pH affect the plant growth. In terms of our hypothesis, it depends on you. What would you like to predict? Remember, with hypothesis, this is an intelligent guess. So maybe for you, once you test before you test those 3 growing medias, you're testing those growing medias, and I just said 3 for example. What are you predicting, or what is your intelligent guess that would happen for example sample A, sample B, C, so forth and so forth, right? So maybe for sample A, you may say it's going to turn out alkaline. For sample B, it's going to be neutral. And for sample C, it's going to be acidic. So no, this is going to be the time for you to write down your predictions. For the predictions, you already written that. Now we're moving on to the materials. At this point, if you're live in the class, you already have those materials with you. If it's a case that you're at home and you're trying to do this by yourself, I want you, as a matter of fact, if you don't have the materials, I want you to go ahead, stop the video or pause the video right now and get them. But once you have those materials, I want you to go ahead and list the materials that you have. In this case, I just only gave examples. So in class, we'll be testing at least 4 grain medias. That's going to be our materials in class. We're going to have distilled water. We're going to have measuring cups. You may be wondering why we need measuring cups. The measuring cup is there for us to measure how many water that we're putting with that medium. All right. We're going to also have our plastic cups or containers, depends on what you want to use, our stirring sticks, or spoons, a pH meter, or the pH testing strips. We're going to have our notebook or a chart and the labels and markers. And one thing I forgot to list on here, the type of plant that we'll be using at that time are going to be beans. So we would already have planted these plants. We're just collecting a sample from each of those beans, depending on the grain media that we're using to grow the beans in. So we would have that in advance. The variables, no, while you're working on that experiment or even after, that's where you're going to figure out what variable is which. So variables, independent, and I put the examples on here. For independent variables, we're looking at the type of grain medium that you're going to use. The dependable, we're going to look at, I did I say definition? I mean, what exactly you're looking for at these areas. For the dependable variable, we're looking at the measure of the pH level, and then our control variables, we're looking at the amount of grain media used, the amount of distilled water, the mixed time, and the testing method. For procedure, is the procedure right here is telling you step by step exactly what you're going to do for your test so you can have your results. And once you have followed the instructions keenly and to the point, then we can move on to our next part. So by now, if you already have done the test, you can actually put your results. So you remember early in the lesson, excuse me, we're looking at texture. We talked about texture of different grain medias. So depends on what you have on hand, and even in the classroom, depends on what we have on hand. That's what we're going to test. So if you notice, I have sample A to D. You can either name them from A to D or actually have their names. Then you're going to tell me what the color of that sample is with their texture. Once you have that, of course, before the experiment, you need to give me a prediction of the pH. What do you think the pH of sample A is going to be? Is it going to be from maybe 5 to 8? Or to be more accurate, say it's 5. Maybe B is going to be 15. This is your prediction because we haven't talked about pH yet. So it's going to be your prediction. And then we're going to see the outcome of that measured pH. And from there, before you actually get to fill this out, we're going to talk about the pH lesson right after that. So you can actually determine based on your result for the measured pH whether it's acidic, neutral, or alkaline. Now, of course, what you observe throughout, and you're going to write that. And of course, as I said before, once you do this part, we're going to pause this lab and then we're going to talk about the pH, what pH is. And then you have your conclusion questions that you're going to answer based off your results. So now we're going to go into nitty gritty of understanding and interpreting our pH test results, as we have already by now looked at our results for growing media samples. So the standard is still the same. We're demonstrating greenhouse crop production techniques, right? And we're looking at standard 5, objective 5, which we're going to investigate the chemical characteristics of growing media. In this case, we're going to interpret our test results. But before we can actually interpret, I can read and interpret the pH test results of growing medias. And you also want to be able to define. So first things first, before we can interpret, we need to know what pH is. So when it comes down to pH, think of it this way. If you look at this scale, let me turn on my laser. If you look at the scale, this is known as a pH scale. Tell me, what do you notice about this pH scale? It might be the colors, right? You may notice it says acidic from 0 to 6, neutral at 7, alkaline from 8 to 14. And that's exactly what it says. So when it comes on to the pH, or the pH scale in general, it's going to measure how acidic or alkaline, in this case, the growing media is. So as you can see, the scales were the range. So be more specific, acidic is going to be from 0 to 6.9. Alkaline is going to be at neutral, right there, dot on the dot for neutral, sorry. And then alkaline is going to be 7.1 to 14. With the pH scale or meter, some people use a pH scale strip or they use a meter. It helps to test and also read the pH based off the color or the digital number that is seen in front of you. Reason why I want to do this is to be able to understand what is going on with our soil so we can understand and be able to help our plant to make sure they're at their ideal pH range for plant growth. Because remember, if that plant has this accurate pH rate, then, if and only then, it's going to absorb the nutrients that it needs in order to grow and thrive. It's going to use all the materials that is being provided from that growing media to be at its best, to be at its peak. So with most plants, the pH is going to be between 5.5 to 6.5. I know someone might be asking, well, if the pH is okay, do I necessarily need to add nutrients to my growing media? Yes, you do. You do. Okay? Not because the pH is good means that you don't add anything afterward. You need to add some nutrients, especially in a hydroponic system. You're going to need those nutrients because remember, with a hydroponic system, the nutrients they're there but they're low. So when you are providing more nutrients to your plants, you're going to do what it needs to do, right? However, we want to also make sure that pH is accurate for that plant to absorb the necessary nutrients that it needs. And that's going to help with the plant health and of course with the root health as well. All right, so now let us look at the different types or different ways we can read pH. So we have our pH monitor and we also have our pH strips. A lot of times, most people may use the pH strips, which are located on the right side of my screen. Other times, people may use the pH meters. So let's look at the differences between them. When it comes on the pH meter, as you can see from this picture, that they are digital. They're going to give you readings, digital readings. With a pH meter, it's going to be paper. So you're looking at, once I dip this paper in, I have a chart and I'm going to see what color matches with that paper. Sometimes it don't even have to be a paper. Like at school, we have a container that collects the sample. Once we have that sample collected, we drop the pH solution in there. And then once it's mixed in with the pH solution with that sample, you have to compare it to the chart, which is practically similar to the strip that you're seeing right here. Now, when it comes onto the digital meter, it's going to be higher. It's going to be more accurate and precise just because it's based off units. While the strip is more of a moderate, it's just giving you the range based off that color, right? Of course, if you're using anything digital, you have to collaborate it. You have to make sure once it's collaborated, it's at its accurate reading. When it's like a strip, you don't have to do that. That's more of a digital thing. The ease of use, people tend to find a hard time using these. I know it seems weird, but at the same time, if you don't take proper care of it and maintain it, it's not going to do what it's supposed to. While the strip is very easy, dip, you dip, you read the color, one time use, boom, done with. If it's a strip, as I said. If it's like what we use at school, with technically the chart is reusable, so you still use a chart. But always we just throw away the sample with the solution, rinse it out. Once it's cleaned properly, start over again. With a strip, it's just a one-time deal. The cost, of course, because it's digital, is going to cost more. With a strip, it's cost less. In terms of usage, it's very ideal to use the pH meter for lab experiments and repeat experiments, while a strip is great for quick readings and class demonstration. So quick checks like hydroponic system and class demonstrations. Durability, as I mentioned before, with the meter, you're going to use it. You can use it multiple times as long as it's taken care of real well. And for the strip, it's a one-time deal, one-time use. All right. So by now, since we have gone over this unit, if if you go back and review the notes on the pH, the pH scale, and by now you may have these on hand. If not, you may have to buy it for your experiment. Depends on where you are. Know what you could do with this information based off your results. You can compare and contrast the pH scale with your results and see which range your growing meter is at. So you can determine if this may be the right environment for your plants. And of course, if you go back to your conclusion questions, it's going to give you a hint of how to answer or come up with your answers for your conclusion. Thanks for listening, and I hope this helps if maybe when you're in class and I was explaining it was not the best way of you grasping the concept. This may have been helpful. I would definitely, I know this is a very long video, so maybe you're watching a long video to this, or maybe you're seeing small chunks of this video. You have a wonderful day. I'm glad that you learned a lot about growing medias in this wonderful unit, and be blessed.